final electron acceptor, meaning it needs to give this away to a different molecule. All right, so for an aerobic organism, that final electron acceptor is going to be oxygen. Oxygen will be the final electron acceptor for an aerobic organism. And water would be produced as well in that scenario. There, now you can see it. There are some organisms that are anaerobic. So that means that they don't live in the presence of oxygen, and the reason for that is because they don't use oxygen as a final electron acceptor here. Instead, they're going to use a different molecule. So for example, some anaerobes will use hydrogen or hydrogen sulfide and use that as their final electron acceptor, and they won't make water. They'll make other different byproducts like methane gas or hydrogen sulfide gas or something like that. So those, again, are anaerobes that don't use oxygen as a final electron acceptor, so they don't live well in the presence of oxygen. Okay, let's see. Nitrate can also be used as a final electron acceptor, and then nitrite is produced. So there's a lot of variations here. And you'll learn more about that and hear that again as we talk in later chapters about some of the characteristics of different bacteria. Okay, so moving on. Let's go back to this. Why are all these enzymes passing this electron down and doing these redox reactions? The reason is that these redox reactions will power the active transport of hydrogen against its concentration gradient to make a concentration gradient outside of the membrane. Okay, so let's go through that again. These redox reactions are pumping hydrogen ions against their concentration gradient. They're moving them all outside of the cell, and it's making a concentration gradient, so a high concentration of hydrogen ions outside of the membrane. So this is an active transport mechanism that requires energy because it's against their concentration gradient. All right, so why did they do that? Well, this box at the end, you may be wondering what this is. This is an enzyme called ATP synthase. ATP synthase structurally allows hydrogen ions to diffuse passively down its concentration gradient, and when it does that, it spins this turbine-like structure, and when that happens, it phosphorylates ADP into ATP. Okay, so these hydrogen ions are going to diffuse passively down their concentration gradient through this ATP synthase, and that's going to power a structural mechanism that is going to generate ATP. And how much ATP? 34 ATP if the conditions are right. So that is going to produce the most ATP out of any of these other steps. All right, so we're going to talk a little bit more about these. We have several different steps here on how ATP is produced. And these can be classified in two different ways. And you'll learn about a third way when you learn about photosynthesis. So these first two ways that ATP are produced in glycolysis and in the citric acid cycle, what happens is ADP, so remember ATP has the adenosine molecule, and then it has the 3 phosphate molecules that make a tail. So These ATP are made when ADP, so there's only 2 phosphates on ADP. Basically, to make the ATP, it just takes a phosphate from another molecule and sticks it on to make the third phosphate in the tail. So that's called substrate level phosphorylation, where it just takes the phosphorus from a different molecule and sticks it onto ADP. Oh, I'm sorry. That's substrate level phosphorylation. So this ATP is generated by substrate level phosphorylation, and this ATP is substrate level phosphorylation. All right. So whenever this ATP is made in a different way, whenever there's a concentration gradient that's used to power the ATP synthase to generate ATP like this, whenever there's a concentration gradient used to make ATP, it's called chemiosmosis. And there's 2 types of chemiosmosis. I ran out of room here. I have to erase some. Okay, so whenever there's a concentration gradient used to generate ATP, that's called chemiosmosis. If there's no light required, like in the electron transport chain, that's called oxidative phosphorylation. So this ATP was produced by oxidative phosphorylation. If a concentration gradient is used but light is required, that's photophosphorylation. That is not used in carbohydrate catabolism, but it is used as a mechanism in photosynthesis. So photophosphorylation is for photosynthesis. Oxidative phosphorylation is how this ATP was generated in the electron transport chain. So oxidative phosphorylation. Alrighty, so the next thing we're going to talk about is where these occur in the cell. So I need to erase our board to do that. Alrighty. This is my eukaryotic cell. This is my prokaryotic cell. Remember, in a eukaryotic cell, there are membrane-bound organelles, and one of those is a mitochondria. I'm going to draw a really huge mitochondria. And remember that the mitochondria have 2 membranes. If you remember back to endosymbiotic theory, they have 2 membranes. And the inner membrane is folded. So that's mitochondria. That can be our nucleus. All right, so, and remember prokaryotes do not have membrane-bound organelles, so I didn't draw anything in this one. All right, so for both prokaryotic and eukaryotic cells, glycolysis occurs in the cytoplasm. Here, I'll write that in blue. So glycolysis is occurring in the cytoplasm. The prep step in a eukaryotic cell will occur in the inside of the inner membrane, and that area is called the matrix. So I put the matrix and I put a P in there for the prep step. For a prokaryotic cell, the prep step also occurs in the cytoplasm. All right, for a eukaryotic cell, the citric acid cycle will also occur in the matrix. So I put a C for that. It's in the cytoplasm in a prokaryotic cell. All right, the electron transport chain requires a membrane. So the electron transport chain will be embedded in that inner membrane in the eukaryotic cell. So I wrote that along that inner membrane. And then remember how an ion gradient is formed? That's going to be pumping those hydrogen ions kind of between the membranes, and they diffuse back into the matrix when they flow through the ATP synthase. In a prokaryotic cell, there needs to be a membrane, right? Because the enzymes are embedded in the membrane. So simply just the cell membrane is used to house the electron transport chain. So that's just occurring in the cell membrane. All right. Next, we need to talk very briefly about fermentation. So in fermentation, sorry, I was going to look for my other marker, but I suppose we don't have it. So like I said, in fermentation, we don't have all of the steps that carbohydrate catabolism does. We only have glycolysis. And I suppose I don't need to write the rest of the steps here, but after glycolysis, there can be different ways that an organism ferments, and different products will be produced depending on the type of fermentation that they produce. So you can have different products, like lactic acid can be a product. You can also get ethanol and carbon dioxide, like you observe with beer brewing, and things like that. With lactic acid, that's used a lot in food production, so think about the taste in sour cream or the tang in different yogurts and things like that. And there's many other byproducts that can occur depending on the type of fermentation that occurs. Essentially what happens in fermentation is after glycolysis, after those molecules of NAD get reduced, those NADH molecules then get oxidized and then are recycled back to glycolysis to be reduced again. So there's kind of this recycling of those electron carrier molecules. All right, so I think that's it for carbohydrate catabolism. So I'll end this lecture now.